The standard explanation says Neanderthals were driven to extinction by climate collapse, warfare, or disease roughly 40,000 years ago. Ancient DNA and population modeling tell a very different story. At their peak, as few as 5,000 Neanderthals may have occupied all of Eurasia, scattered in bands of around 50. A 2.7% drop in fertility among first-time mothers was mathematically sufficient to trigger their disappearance within 10,000 years. No catastrophe required. The real question is how a lineage that survived over 300,000 years became that vulnerable in the first place. Archaeological site counts and genomic analyses converge on a strikingly low Neanderthal census. Estimates place the total population across Eurasia between 5,000 and 70,000 individuals at their peak. Most lived in small, scattered groups known as bands, each numbering around 50 people. These bands operated with limited contact, separated by vast distances and harsh terrain. Effective population size, a genetic measure reflecting how many individuals actually contributed to the gene pool, rarely exceeded 3,000 to 5,000. Such a figure is an order of magnitude below the census population, underscoring how few Neanderthals were breeding at any one time. Site density metrics reinforce this picture. Occupation densities in core regions like Iberia and the Levant rarely surpassed 0.02 individuals per square kilometer. In many areas, densities dropped below 0.01 individuals per square kilometer, indicating that entire valleys or mountain ranges could be home to a single band or none at all. The archaeological record supports these numbers. Radiocarbon-dated sites show sparse occupation, with many regions yielding only a handful of contemporaneous settlements. Ancient DNA studies from the Altai and Vindija Neanderthals show long-term genetic isolation and low diversity, matching the expectations for fragmented, thinly distributed populations. Demographic models based on these inputs reveal a system operating near the minimum threshold for persistence. With so few individuals per band and so little gene flow between them, random events such as accidents, disease outbreaks, or even a bad season could erase entire communities. The metapopulation structure, with isolated bands occasionally exchanging mates, offered some buffer against total collapse, but it also amplified the impact of local extinctions. A band that failed to replace itself could not be easily rescued by neighbors, especially as overall densities declined. These parameters, small census size, low effective population, and sparse site density, created a demographic environment highly sensitive to even minor disruptions in birth rates or survival. No catastrophic event was needed. The numbers alone set the stage for vulnerability, making Neanderthals acutely vulnerable to demographic shocks. In 2019, Anna Degioanni applied a Monte Carlo simulation to probe the demographic stability of Neanderthals. Her approach was grounded in numbers. She ran 10,000 extinction scenarios for populations ranging from 5,000 to 70,000, using fertility and mortality rates drawn from modern hunter-gatherer data. The baseline was clear. If each woman had an average of 2.6 children, the population could persist for over 30,000 years. But the model's power lay in its ability to isolate the influence of a single demographic variable, the fertility of young, first-time mothers known as primiparous women. A seemingly minor reduction, just 2.7% fewer births among these young mothers, caused the median extinction time to plummet to about 10,000 years. Pushing that reduction to 8% brought collapse in only 4,000 years. The effect was not gradual or linear. Instead, the smallest changes in the reproductive output of the youngest mothers rippled through generations, compounding over time. The result? A slow, relentless decline toward extinction occurred even in the absence of external shocks like climate change, disease, or conflict. Degioanni's model did not require catastrophe. It showed that demographic fragility alone could explain the disappearance of Neanderthals. Sensitivity analyses reinforced this conclusion. The system was most vulnerable to changes in primiparity. Increases in juvenile mortality or longer gaps between births had some effect, but nothing matched the impact of reduced fertility among young mothers. The population, already near its minimum viable size, could not absorb even modest, sustained drops in this key demographic group. The simulation's 10,000-year extinction window closely matches the archaeological record, which places the disappearance of Neanderthals between 41,000 and 39,000 years ago. Degioanni's findings quantify how internal demographic mechanisms, rather than dramatic external events, can drive a species to extinction. The numbers reveal a system on a knife's edge, where the fate of an entire population could hinge on the reproductive success of its youngest women. This insight reframes the search for causes. Before considering genetic or cultural contact with Homo sapiens, we must ask whether Neanderthal decline was already underway, set in motion by the quiet mathematics of fertility and survival. In 2010, Richard Green and Svante Pääbo published the draft sequence of the Neanderthal genome, assembled from more than 4 billion DNA fragments taken from 3 individuals. Their analysis revealed that non-African modern humans carry between 5 and 6% Neanderthal DNA, a signal consistent across European and Asian populations. The admixture event was dated to between 55,000 and 65,000 years ago, based on the length and distribution of Neanderthal-derived segments in ancient and present-day genomes. The initial pulse of gene flow left a measurable imprint. Every non-African individual alive today carries multiple stretches of Neanderthal ancestry scattered throughout their genome. This finding closed a decades-long debate over whether Neanderthals and Homo sapiens ever met and bred. The data showed unequivocal contact, with gene flow occurring soon after modern humans left Africa and spread into Eurasia. The Green and Pääbo team used stringent contamination controls, including damage pattern analysis and independent replication, to confirm the authenticity of the sequences. The draft genome covered approximately 1.3% of the human reference sequence, sufficient to detect introgression peaks in genes related to skin, immunity, and cognition. Subsequent studies refined the admixture estimate but confirmed the essential result. Neanderthal DNA persists in living humans, particularly outside Africa. This large-scale ancient DNA sequencing project established the technical foundation for later work on hybrid fitness, genomic incompatibilities, and the evolutionary consequences of interbreeding. The presence of Neanderthal ancestry in modern genomes opened the door to investigating how these ancient segments affected health, reproduction, and adaptation in subsequent generations. In 2016, Fernando Mendes and his team sequenced the Y chromosome of a 49,000-year-old Neanderthal from El Cedron Cave. Their analysis identified 3 damaging mutations in genes essential for spermatogenesis: KDM5D, PCDH11Y, and USP9Y. The KDM5D gene carried a nonsense mutation truncating its catalytic domain, a change known in modern humans to cause severe oligozoospermia and Sertoli cell-only syndrome. In PCDH11Y, a missense mutation altered an extracellular cadherin repeat, disrupting cell adhesion in developing sperm. USP9Y showed a frame shift deletion that eliminated its enzymatic domain, a lesion linked to complete azoospermia in human patients, and infertility in knockout mouse models. All 3 genes are part of the azoospermia factor region, expressed exclusively in testicular tissue. Functional loss in these genes impairs sperm maturation and viability. Mendes's findings suggest that Neanderthal Y chromosomes encoded male-specific proteins that differed enough from modern human variants to trigger immune recognition. The HY antigen, a minor histocompatibility protein encoded on the Y chromosome, can provoke maternal immune responses when sufficiently divergent. In hybrid pregnancies, modern human mothers exposed to Neanderthal HY epitopies may have mounted T-cell responses leading to miscarriage or failed implantation, particularly for male embryos. This mechanism is supported by clinical studies in humans where anti-HY antibodies are associated with recurrent miscarriage and male infertility. Comparative work in mice and primates shows that even subtle Y-linked antigenic differences can result in immune-mediated hybrid sterility or fetal loss. The presence of these 3 disruptive mutations provides a direct biological pathway for reduced hybrid male fertility. It also offers a mechanistic explanation for the complete disappearance of Neanderthal Y chromosomes from modern human populations. The HY antigen hypothesis links molecular incompatibility to demographic outcomes, showing how sex-specific reproductive barriers could have amplified the vulnerability of Neanderthal lineages during periods of contact and admixture. Martin Peter and Janet Kelso published a 2020 analysis that examined the fate of the Neanderthal Y chromosome in modern populations. Their team compared ancient Neanderthal Y sequences to a dataset of over 2,500 present-day male genomes. Not a single individual carried a Y chromosome haplogroup traceable to Neanderthals. This result stands in contrast to the widespread presence of Neanderthal DNA elsewhere in the genome, where 1 to 2% persists in non-African populations. Pater and Kelso tested whether genetic drift alone could explain this absence. Simulations showed that even under the most conservative assumptions, a complete loss of Neanderthal Y chromosomes from the gene pool by chance was extremely unlikely. The data best fit a scenario where selection, rather than neutral processes, drove the Neanderthal Y to extinction. 2 models were considered: a selective sweep of modern human Y chromosomes through repeated male-biased gene flow, and purifying selection against Neanderthal Y-linked incompatibilities. Both models predict rapid replacement in small, fragmented populations. The findings confirm a sex-specific pattern of genetic loss. While autosomal and mitochondrial Neanderthal lineages survive in modern humans, the male line was erased entirely. The absence of Neanderthal Y chromosomes in every surveyed modern male underscores the impact of reproductive barriers and selective processes at the population level. This outcome provides direct genetic evidence for the complete turnover of paternal lineages during the late Pleistocene, setting the stage for broader analysis of genome-wide selection against Neanderthal ancestry. In 2014, Sriram Sankararaman and colleagues published a genome-wide map of Neanderthal ancestry in present-day humans. Their analysis used a hidden Markov model to scan thousands of modern genomes for Neanderthal-derived DNA segments. The results revealed a striking pattern. The X chromosome carried far less Neanderthal ancestry than the autosomes. While most chromosomes retained 1 to 2% Neanderthal DNA, the X chromosome held less than 0.2%. This depletion was not random. Regions of the X chromosome that overlapped with genes expressed in the testes showed the strongest reduction. The odds ratio for introgression in these testis-expressed genes dropped to 0.25 compared to the genome average. The statistical significance of this pattern exceeded p less than 1 times 10 to the minus 6 after correcting for background selection and local recombination rates. The data indicate that purifying selection systematically removed Neanderthal DNA from regions critical to male fertility. The effect was not limited to a handful of loci. Instead, it extended across large stretches of the X chromosome, creating deserts of Neanderthal ancestry. These deserts often spanned several megabases and included genes involved in spermatogenesis and meiosis. Sankararaman's team compared the depletion to patterns seen in other mammals, where hybrid male sterility is often linked to incompatibilities on the X chromosome. The findings fit Haldane's rule, which predicts that the heterogametic sex, in this case males, will suffer reduced fertility in hybrids. Additional analyses showed that the depletion was strongest in populations with the highest initial Neanderthal introgression, supporting the role of ongoing selection. The study used high-coverage genomes from the 1000 Genomes Project and the Altai Neanderthal as a reference. The hidden Markov model detected Neanderthal tracks by their sequence divergence and linkage patterns, allowing precise mapping of introgressed segments. The overlap between introgression deserts and testis-expressed genes was confirmed by cross-referencing gene expression databases. The genome-wide pattern of selective pruning provides direct evidence that incompatibilities in reproductive genes played a major role in shaping the fate of Neanderthal ancestry. The systematic removal of these segments did not require catastrophic events or rapid extinction. Instead, it points to a prolonged process of purifying selection, acting over thousands of years, gradually reducing the Neanderthal contribution in functionally sensitive regions. These findings set the stage for mathematical models that quantify how such selective pressures, combined with demographic dilution, could erase distinct Neanderthal lineages from the modern gene pool. Andrea Amadei and Simone Fattorini published a mathematical framework in 2025 that quantifies how Neanderthal genetic identity could be erased without catastrophic events. Their dilution model treats each Neanderthal group as a closed island population, receiving a steady trickle of Homo sapiens migrants. The core equation reads, the derivative of p with respect to t equals minus mu times p plus m times 1 minus p. It describes the changing proportion of Neanderthal ancestry, p, over time, with mu representing loss from genetic drift, and m the migration rate per generation. Using archaeological and genomic data, they set m at 0.5% per generation. With this value, simulations predict that 99% of Neanderthal genetic material would be replaced within 10,000 to 30,000 years. The process is gradual, requiring no selective advantage for either group. Even in the absence of warfare, epidemics, or climate crises, the sheer demographic disparity between Neanderthals and incoming Homo sapiens is enough to drive near total replacement. The model starts with a pure Neanderthal population and applies a constant migration rate. Each generation, a small fraction of the population is replaced by newcomers. The effect compounds over time, with the Neanderthal proportion shrinking exponentially. Monte Carlo simulations confirm the analytic solution. At 0.5% migration per generation, fewer than 1 in 100 Neanderthal lineages persist after 1,000 generations. With a generation length of 25 years, this matches the archaeological disappearance window. The model's outcome does not depend on environmental collapse or violent conflict. Instead, it demonstrates that Absorption into a larger, more connected population can erase a distinct lineage through demographic and genetic processes alone. Amade's results align with observed patterns in ancient DNA. The highest Neanderthal ancestry is found in the most isolated groups, while regions with greater contact show accelerated dilution. The disappearance of Neanderthal Y chromosomes and mitochondrial lineages fits the same framework. Replacement occurs genome-wide, not just in specific markers. This synthesis integrates demographic fragility, genetic incompatibility, and sustained gene flow into a single quantitative explanation. The evidence supports a scenario where Neanderthals did not vanish in a single event, but were gradually absorbed over thousands of years, leaving only fragments of their DNA in modern populations. The dilution model accounts for the scale and timing of Neanderthal extinction, showing that demographic and evolutionary forces, operating over millennia, can explain the near total replacement of one human lineage by another. Neanderthal extinction models now center on demographic thresholds and gene flow, not apocalyptic events. Population genetics confirms their DNA persists in over 40% of living humans. The lesson is clear. Extinction can unfold invisibly as numbers decline and lineages dissolve through gradual absorption. As global biodiversity shrinks and small populations face similar risks, the Neanderthal story warns that survival hinges on more than strength or intelligence. Sometimes it is just math,